This protocol facilitates the identification and isolation of live coral cell populations at a descriptional level not previously possible. The main advantage of this technique is that it allows access to a level of cell specificity that up until now has been mostly unattainable. Demonstrating the procedure will be Dr. Shannon Say, a research associate from the flow cytometry shared resource at the Sylvester Comprehensive Cancer Center.
To set up the flow cytometer for live coral cell sorting, open a new project template in the flow cytometer software and select the appropriate lasers for the analysis in the laser panel. Then, create a side scatter versus forward scatter plot in the appropriate plots for setting up the gating and analysis strategy. For flow cytometric analysis and sorting of the live coral cells, load the control unstained cell sample into the sample chamber of the cytometer and start the reading process.
Since coral cells are particularly fragile, keep the cytometer pressure low to prevent cell lysis.
In the scatter plot, adjust the photomultiplier voltage to center the points and begin recording the events. In the forward versus side scatter plot, create a gate around the cells at around the 10 to the 4th mark on the forward scatter x-axis. Replace the control sample with the first sample of stained cells and record approximately 10,000 cells before pausing. Then gate the events that are unique to the stained sample group. To sort live coral cells, after selecting the cell population of interest in the flow cytometer software,